Hello, hello everyone. Nivedita is here, taking a new video on the topic transcription in eukaryotes. Okay, this is a topic from class 12th biology, and the chapter is 6th molecular basis of inheritance. This is transcription. Before going to start this topic, transcription again, I'm telling you, transcription means formation of RNA from the DNA. Okay, this is called as the transcription. Central dogma you should know. Okay, central dogma gives the information in the molecular biology. So this is one of the flow of information. Okay, though transcription in prokaryotes and eukaryotes, it is different, though the earlier process is similar in both prokaryotes and the eukaryotes. What is different in the eukaryotes from the prokaryotes, that is explained in this video. Okay, initial processes are same, like unwinding and all. Yes, the central dogma of molecular biology. So this central dogma was proposed by Crick, and this states that genetic information flows unidirectionally, means from DNA to RNA and RNA to protein. Means keep one thing in mind, that the retroviruses, they do not follow central dogma because information is not unidirectional there. So this is very important one. The flow of information should be in the one direction. So this happens in the process of protein synthesis. That is, protein synthesis, how protein synthesis is having two main steps: transcription and the translation. Okay, formation of RNA from the DNA is transcription, and formation of or synthesis of protein from the RNA, that is translation. Okay, now how the transcription in eukaryotes, it is different. So it begins from here. In eukaryotes, the structural genes are monocistronic, whereas there in the prokaryotes it is polycistronic, and there it is continuous, but here in the eukaryotes it is a split. Split means discontinuous, means mRNA just consists of some non-coding parts as well, segments as well. Okay, so the coding sequences are called as exons, okay, which will form the part of mRNA, and the non-coding parts which interrupt in between the exons, that is called as introns. So introns are not the part of mRNA and they need to be removed, and their removal is called as splicing. Okay, so this splicing occurs in eukaryotes only, not in prokaryotes. Okay, there the mRNA, that is continuous one, no interruption. So that's why there is continuous, but here it is split. Okay, so before it is translated, there will go on splicing. So this is most important part, which is not required in the prokaryotes one. Again, there is difference here in the eukaryotes. There participate three different types of RNA polymerase, whereas in the prokaryotes only one type of RNA polymerase is able to make all type of RNA. Okay, so in eukaryotes there are three types of polymerase which will participate in the transcription. So RNA polymerase 1, that is for rRNA. RNA polymerase 2, that is for hnRNA. Now question is, what is hnRNA? Heterogeneous nuclear RNA. Okay, heterogeneous nuclear RNA, that is synthesized, or that is formed by the enzyme RNA polymerase 2, and RNA polymerase 3, this is for tRNA. Okay, so there are different types of RNA polymerase for different types of RNAs. Now what is hnRNA? hnRNA, I told you, heterogeneous nuclear RNA, means this is not the matured RNA. Okay, this needs the maturation. This will not work. Why this will not work? Because what happens, this hnRNA, this contains both exons and introns. Already I told you, exons are expressible parts and introns are not the expressible parts. Okay, so it will require the splicing. Means splicing is, in the process of splicing, introns are removed and the exons are joined together. So when all the introns are removed, then forms the mRNA, messenger RNA. That time it forms, means the hnRNA converts into mRNA, and this is called as processing. This step, whole step is called as the processing. More are there, still the process is continuous. Now what happens after splicing? The RNA undergoes two additional processes called as capping and the tailing. Capping and the tailing, without it, it is not activated one. So in the capping, what happens? Methyl guanosine triphosphate, this is added, this group is added, methyl guanosine triphosphate. Okay, this is added to the 5' end of hnRNA. Okay, so when this group added at 5', this is called capping is done. Tailing 1. What happens? Capping means it is at 5' end and the tailing is at 3' end of hnRNA. So what happens? There the residues, adenylate residues, 200 to 300 adenylate residues, these are added at the 3' end, and now this whole is the processing of hnRNA, and completely hnRNA is converted into mRNA, and this will be released from the nucleus. Okay, because this is formed in the nucleus only. So this whole step, splicing, capping and the tailing, these are the parts of processing of hnRNA. These are the diagrammatic representation of the process of transcription in eukaryotes. Here you can say the first step is the common one, that forms their one of the strand act as the template. Okay, where which will form the complementary mRNA, and what is formed there, RNA, what is formed over there, you can see all the introns are removed and exons are joined and capping is shown. Okay, so at the end what happens, the hnRNA is converted into the mature RNA, mRNA.